The Nautilus platform is designed to conduct two kinds of analysis: proteoform analysis, covered here, and broad-scale discovery proteomics, covered in our video titled Quantifying Proteins on the Nautilus Proteome Analysis Platform. In this animation, we cover what proteoforms are, quantifying proteoforms on the Nautilus platform, and applications of proteoform analyses. What are proteoforms, and why are they important? Proteoforms are any variants of genetically encoded proteins. While the genome specifies the sequences of amino acids that make up full-length proteins, these sequences can be modified in many different ways, a few of which include alternative splicing, which can remove or join different sections of a protein; phosphorylation, which adds phosphate groups to proteins and may alter their activity in a variety of ways; ubiquitination, which adds ubiquitin to a protein and can, among other things, target a protein for degradation. As you can see, different proteoforms can have vastly altered or modified functions even though they are encoded in the same gene. Thus, understanding proteoforms and their abundance is key to understanding the role of proteins in cellular function and, consequently, health and disease. Quantifying proteoforms on the Nautilus Proteome Analysis Platform. We start by depositing single protein molecules from either the full proteome or a select subset of the proteome onto up to 10 billion distinct landing pads. Then, using proteoform-agnostic probes, we identify all the landing pads occupied by the protein species of interest. Later, using either probes specific to a particular proteoform or a particular type of modification, we identify which proteoforms are found at each landing pad occupied by the protein species of interest. Because each landing pad contains only a single protein molecule, we can count the number of times a proteoform is identified to determine its abundance. At the single molecule level, identification equals quantification. On other platforms, it is often impossible to distinguish separate proteins modified in single but distinct ways from single proteins modified in multiple ways, with each scenario having potentially different functional consequences. Because our platform analyzes intact proteins at the single molecule level, these different proteoforms are readily distinguishable. Applications of proteoform analyses. There are many reasons to study proteoforms. We highlight discovery research and translational research here. First, studies show that different tissues express different proteoforms. To gain a real understanding of how tissues function when healthy, we must acquire a far more in-depth understanding of proteoforms. Second, aberrant proteoforms play important roles in diseases like Alzheimer's, where hyperphosphorylated tau protein is correlated with detrimental neurological effects. Having a more in-depth understanding of proteoforms present at different stages of disease may help us develop new means of diagnosing diseases earlier or treating them through proteoform-targeted drugs. Overall, proteoforms exponentially expand the already vast landscape of the proteome. Being able to comprehensively quantify proteoforms will provide many more routes of discovery that will improve basic research, healthcare, and more. Learn more at nautilus.bio.